which is much like M-fold in that it implements the algorithm of Zucker and Stiegler, or McCaskill's algorithm, in order to find minimum 3 energy structures for our Terminator hairpins. And we also use a program called Kinfold, which is a Monte Carlo simulator that simulates RNA folding in terms of the secondary structure at an elementary step level, which is to say that you can bond to nucleotides, you can unbond to nucleotides, or you can switch partners with some available region of the RNA, as long as there are no pseudoknots, and this is entirely with respect to secondary and not tertiary structures. One issue we had, however, was that Kinfold does not have a real-time unit on it. It uses some arbitrary simulation step that we had to calibrate. And in order to do this, we used the results from Li-Sauerwine in 1998 to fold a certain hairpin, and by comparison of the time that Li-Sauerwine got the folding to the time that we got the folding using Kinfold, we could infer that the Kinfold time step must be about 5 microseconds. And this is important because for the remainder of the talk, I will often speak of the Kinfold grow time, which you can think of as an inverse growth rate. In other words, if I say there is a Kinfold grow time of 4,000, each nucleotide will take 4,000 Kinfold steps to transcribe, which corresponds to, in the case of 4,000, the biological relative grow time of 50 nucleotides per second. So we had 2 important control groups. One was to take our biological Terminator hairpins and grow them in the 3' to 5' direction, as opposed to the normal 5' to 3' direction. This is a good control because we're guaranteed to get the exact same minimum free energy topology and the exact same energy, therefore, for that topology, except we're most assuredly getting different folding paths to that final folded hairpin state. The issue here being that with reverse growth as a control, we only get as many controls as we have biological hairpins, and we just don't have very many of them because of the time involved in locating these on the RNA. Another control we used was to randomly shuffle the nucleotides in the sequence, but ensure that as we shuffle the nucleotides, we will end up with a hairpin topology. So, of course, we can think of many different ways to shuffle the nucleotides in the hairpin, but we chose the one that gives the strictest bound on significance of our results. But the other thing to note is that qualitatively there wasn't a really big difference between all the different types of shuffle. So I'll soon be showing the results, and the plots you'll see, the first set at least, will look something like this. This is for the YXJD gene in Bacillus subtilis. And what you see here is the average proportion of the minimum free energy structure found as a function of the growth time in Kinfold, or time per nucleotide transcribed. So the black line is the actual forward transcription from 5' to 3' of the logical hairpin. The red line is the average NFE structure found when growing the biological hairpin in reverse. So it's noteworthy that in this case, the reverse hairpin performs worse than the forward hairpin up to a certain time. And then you see the behavior of our random population in forward and reverse growth. So there wasn't a big difference in forward and reverse growth, mostly because for each random hairpin in our random population, its reverse is also numeric. So we would expect the forward-growing sequence of nucleotides to perform similarly to the reverse sequence folding also from 3' to 5'. So this isn't too surprising. Any individual random hairpin is likely to exhibit some form of reverse asymmetry, but it's washed out. So this controlled growth time 4000 is the actual bacterial growth rate taken from E. coli, and anything you see to the left corresponds to the hairpin being growing faster than 50 nucleotides per second, and anything to the right corresponds to slower. So keep that in mind when you look at this next set of plots. Here I've labeled for you the average proportion of the NFE structure found on the vertical and the growth time on the horizontal, but you can focus on growth without the biological growth rate. So notice that in each case, the real hairpin performs better than the random set. And in this case, it performs dramatically better than even the random set as well as the reverse. So we have the same story for FMN, Bacillus subtilis, just fewer examples. And the same story for the purine and Streptococcus. And note here that we have the same gene, but in different elements of the Streptococcus family. So one of these for different Streptococcus strains. And here's the same for FMN and Streptococcus. So from these, we can certainly ascertain that the real hairpins seem to perform better than the random population at average. But there's an immediate question here that what exactly, how much better than the random hairpins is this performing? There's something special about our real hairpins. We don't know, since this is an average, the distribution of the random samples along this line. So let me show you for a particular example what you